Right, everyone, it's just hit past 11, so we should get this ball rolling. So hello and welcome to you all who have joined us for this online media briefing on genomic testing and COVID-19. I'm Annika Dean from the Australian Science Media Centre. I'm joined today by Dr. Rebecca Rocket, a virologist at the University of Sydney's Marie Bashir Institute for Infectious Diseases and Biosecurity at Westmead Hospital, and Professor Catherine Bennett, Chair in Epidemiology at Deakin University in Victoria. Thank you both for joining us online today. Today's briefing is one of our Q&A style briefings, so there won't be any presentations from the speakers. We'll just jump straight into questions. To ask questions during today's briefing, you just need to enter them into the Q&A box on the bottom right of your screen. Just make sure to use that Q&A box and not the chat box that sits above us. As this briefing is for journalists, please include your media outlet, as questions from journalists will be prioritised. Please, as I said, ensure you use the Q&A box and not the chat box, and then I will read out each question to our speakers today. For anyone who joins us over the phone, although it looks like we don't have any as yet, we will unmute those attendees later in the event. So I'll kick things off with probably a fairly obvious one that might, if we can start with Rebecca, is just, could you explain what genomic testing is when it comes to COVID-19?
Thanks, Annika, and hello, everyone. Genomic testing is the genetic code or the genetic fingerprint of the virus that causes COVID-19 in this instance. So your genome is something that is quite specific to you. It encodes is the genetic code or recipe that makes all the that encodes all the characteristics about you. And you know, you have a genome, dogs have a genome, and in this instance, viruses have a genome. And we're using really small, minute changes within this viral genome to help kind of piece together how the virus is moving through the community. So the genome itself is around 30,000 base pairs in length. So it's made up of a code of essentially 4 letters. And so the fingerprint is made by particular mutations within that code that happen at very specific places along the genome.
And so how is it that you're able to, what is it that you use genomic testing for? What is it, how does it help us sort of in the fight against COVID?
Traditionally, we've used it for foodborne outbreaks. So when we see a lot of cases of salmonella, the epidemiologist will do some kind of analysis to look at the different types of food people have eaten. And then they will do some environmental swabbing. And what we do, we'll match the genome from the patients with particular salmonella species that could be cultured off fresh fruit and vegetables or chicken, for example. And when we find that match, we can very accurately determine what the source of the outbreak is and then remove that from the supermarket shelves or from the restaurant in question. And so we use that premise when COVID-19 kind of struck in Australia to quickly design this genomic test that would help us kind of group together clusters of patients where we think it was likely that they transmitted the virus between each other or to each other. And that just adds extra evidence to the traditional epidemiology and adds confidence to the kind of mantra at the moment, which is test, trace, isolate. So once you understand potentially how the virus is moving through the community, you can more accurately kind of isolate the contacts of those cases or target your testing strategies to cover particular communities or particular kind of institutions that might be at risk.
So, Catherine, as an epidemiologist, how is genomic testing helping you in your role?
So I think Rebecca's given us a good idea about how important it is to be able to cluster people or cases together that are linked by very closely related, identical almost strains of this virus. So from an epidemiologist's point of view, that's really important because if you're finding lots of cases that appear in the community at once, if you look at the New Zealand situation, for example, then that ability to look at the genome, see that these cases are actually all linked, it tells you you've probably got a point source. You've had, a breach of your quarantine in some way or whatever. And then you can actually link the information about the strain of the virus back to, libraries that are now being built with this bioinformatic information in them that tells us what strains are circulating in different parts of the world. So it actually helps you, trace back some of those quarantine breaches and so on in the big picture. Locally, if you do have variation in different strains circulating, and that was certainly the case in our first wave of the outbreak, we had some 76 identifiable distinct genomic clusters in Victoria at the time because we had so many new introductions from people bringing it in from overseas. It allowed you to make sure that when you looked at close contacts of those people and they tested positive, that you were able to make sure it was the same virus and that there wasn't even more strains circulating in the community we didn't know about. So it gives you this ability to both track back upstream where you know cases are linked, but the other side, which is really interesting from an epidemiologist's point of view, is a bit like Rebecca was talking about the foodborne disease. If you get these isolated cases in the community, and we have a lot of them in Victoria at the moment, if we did have variability in the strains that were circulating in community, it allows you to say, okay, we don't know how these are linked, but if we put them together into a group associated by the strain of virus causing their infection, then that's where we get more bang for our buck. That's where you really want to invest in looking upstream to find common sources, because that's more efficient and more possible once you've teased out separate clusters. If you have a whole lot of different clusters and they're all overlapping, then you get confusing information. You can't look at those patterns to either find or rediscover chains of transmission or do the analytics, which I can talk more about later, but that help you understand what puts those people at risk.
We've heard this week, this could be a question for Rebecca or Catherine Ming in Victoria. We've heard this week about tracing the source of Melbourne's second wave, essentially, including that 99% of it came from hotel quarantine. I guess both from your areas of expertise, how is it that they were able to actually come to that conclusion? Rebecca, do you want to start with the sort of more technical side of things?
Sure. So each of the viruses have this really unique genetic signature. So these are mutations in particular points of the genome, and they're kind of carried on when the virus is transmitted. Obviously, all the cases that were reported in Victoria have a very, very similar genetic signature, and so they can kind of assume that this came from a point source, or not assume, accurately kind of determine it came from quite a point source infection. And that's good news in terms of the fact that they're not dealing on multiple different fronts with different importations of the virus, which Catherine quite accurately described happened in the first wave. So we had lots of people coming from different parts of the world with slightly different viruses. And so we had a lot of importations of these genetic fingerprints, if you like.
So how many viruses do we have, strains of the virus do we have going around at the moment? Are Victoria and New South Wales dealing with the same strains, or are they separate from each other as well?
From the limited data that we have at the moment, they look like they're the same strain. So they've probably returned travellers from Victoria, and that is a good thing in our eyes because we don't want to see multiple strains coming in where we don't understand where the transmission chains have come from. And so a lot of instances you have this kind of recall bias when you do traditional epi. So someone might not remember that they've been at a particular place at a particular time or accurately kind of predict who they've had or who they've had contact with. So I think genomic sequencing adds this layer of evidence that can assist with these cases where we're not quite sure how they acquired their infection. And so at the moment it's being really reassuring to see that, if we do have these breaches, and unfortunately this is a really transmissible virus, then we can quite accurately kind of assess those breaches and then control them.
So just in answer to the question about the 99% as well, it does come back to also how many people are tested, so that what that tells you about what's happening in the community. So we don't test, we know we haven't captured every single case, but also we haven't got genomics on every single case either. And so I think that was really being, scientifically correct and saying we can't be 100% sure that every case currently is linked to the same virus, but we haven't found, as far as I understand it, other genomic fingerprints in the Victorian community. So it's almost a process by exclusion that we're saying, this is in fact caused by a single breach. The COVID virus doesn't mutate as quickly as perhaps some others might. So it's got benefits and disadvantages because you don't have quite the same resolution if you're trying to drill down and look at local clusters, but at the same time, it's stable enough that the virus that came into Melbourne back in June or late May is in fact still identifiable in the New South Wales cases. So it does allow us to look at those incursions and then track the virus in the community over what is now a couple of months, but thousands of transmissions. But we might yet see some other small fingerprint changes, when you just get those random mutations, that also does allow some more drilling down locally as we try and get a better handle on some of the local transmission, which still poses a problem in all our states with these mystery cases.
You've talked about the use of genomic testing and contact tracing, but by the sounds of it, in Victoria at the moment, we're dealing with basically one strain. Does that make it very difficult then to do contact tracing because everyone is linked to everybody?
Well, they're linked to everybody, and we can't use genomics to separate out the clusters. So genomics doesn't add to it. So in Victoria, you can still work upstream. We define our outbreaks in person, time and place. So you're looking for common points with other cases that help you identify a point upstream where they might have all picked the virus up. You might never know who that index case was that gave it to that new cluster, but at least you know where it started and what the risks are about certain environments or certain behaviours or whatever. So we can still do that work. It just means, unfortunately, when people are actually sharing the same strain, whatever branch of the clusters they're in or transmission chains, you can't always find it. Equally, when you do get these isolated cases, and it comes back to Rebecca's talking about how we use it in foodborne disease, that's really important because it does allow you to go back to a common source or a continuing source of infection for community that you can then deal with. Here, when we have individual cases, if we could use genomics to cluster those together that appear to have shared a similar strain, then you can do even more focused analytic studies where you look at what those people have in common. You're not going upstream to find a source, but you're actually seeing what behaviours they have, whether they're on public transport more than others, these sort of case control studies that help us identify what might put them at risk. So we can still do that in Victoria, but we just don't have that genomic resolution because we're sharing this reintroduced but limited range. I think there's 2 strains in Melbourne, and so we can look at that level. But within that, we have so much spread, it's hard to use genomics on the ground in the shoe leather epidemiology.
And Rebecca, in terms of strains, we know there's not many in Australia at the moment, but how many strains are there, do we know, just globally for COVID-19?
I think as a scientist, scientists like to put things in boxes. They like to give things names. And the calling of these different genomic fingerprints as different strains is quite controversial. So overall, when you take a step back, we're trying to drill down to find these minute differences to look at transmission. It's really still just one big strain. We are using the genomics to break it down into what's called lineages. But that's been done by multiple different scientists in multiple parts of the world. And at the moment, considering how quickly this is evolving, there is quite a consensus about how to do that. So there's probably, more than 30 or 40 different lineages. But as Catherine said, in Melbourne, they described 76 clusters. So there's a lot of numbers being thrown around. But I think the take-home message is that this is a really similar strain that's circulating, and there's a lot of questions about particular mutations making the virus more transmissible or more virulent. And I think at this stage, the jury is still out, and there's some evidence that some strains are more transmissible than others, but I think that this virus is very good at transmitting from person to person, regardless of what these small mutations within the genome. But definitely there's a lot of research and effort going into both finding a consistent classification system and looking what these small mutations mean on more of a kind of broader scale about virulence and about transmissibility.
Do we know much about, in terms of the lineage of to use strains now, do we know much about where, in the first wave of the viruses that came into Australia, where was it only coming from one source, or did Australia start bringing it in, I'm assuming from international travellers, did we bring it in from multiple countries, or will we even able to determine that?
Yeah, we, sorry, I've got someone blowing their horn in the background next door. We actually could track the strains or the to different countries, and we also knew from the pattern of travellers where they had come from as well. So it actually changed a bit over time. Early on, we had people coming from Asia, and then we actually had our majority were coming out of Europe and America. So we had a period there towards the second part of the first wave where they were our predominant areas. So the majority were imported cases, but where they were coming from shifted. And then later we had people coming in from, again, the subcontinent, India and places. So it sort of changed with the pattern of Australians returning.
I think sorry, go ahead, Rebecca.
I think as well, early in the infection, I think a lot of, all countries were really interested in sequencing this particular pathogen and getting a handle of what was happening in their own kind of country. And they were uploaded to share these databases that allowed us to look at international strain variation. But I think now we're seeing a lot of sampling bias as well. Like Australia is great at producing genomic data for this bug. So is the US and so is the UK. And I think over half of the sequences that are in databases at the moment are from the UK. So I think as the outbreak goes on, it's actually harder to determine potentially which country of origin this particular strain came from, because you can see, it's almost like you're looking for fingerprints, but you can only match a fingerprint with the fingerprint that's been uploaded. So, if everything's from the UK, of course, you're going to find hits from the UK. So it's great that we're doing so much data sharing, but we just have to be aware of those small biases. And I think most people are fully on board the fact that this isn't about finger pointing. This is about, knowing as much as we can to minimise transmission of this bug.
And also another interesting area, and I think that's a really important point about the bias, just the people putting the information in the library changes what we read out of the library. So it's a very important point. But there have been other interesting, more specific examples, and the one that intrigued me was work they did in America, particularly New York, where they went back over stored sera and they could find, looking at these stored blood samples, different points where it appeared the virus landed in New York. And you can then distinguish whether these are, the same lineages or not, and relate them to the outbreak that occurred where they were, had large numbers of cases and could test it. And so they were able to demonstrate that the virus landed in New York a number of times before it actually took off. And epidemiologically, that's really interesting. It doesn't mean it landed and was sitting there and then took off later. It actually means it landed and died out. It didn't quite meet the right conditions that allowed it to really fly. And that's why we struggle to know exactly where this all started, generally, but also even locally, where it has moved around the world, because it probably landed a few times before it actually, took off. So that's the other way that we can use this fingerprinting to actually try and unpack what went on and understand that a bit better.
We talk about the different lineages. What's actually happening in terms of through each individual person to create the genomic lineages? Does it change as it goes through every person, or can two people have exactly the same looking genomic strain?
So that's a really key area of research, why we see particular clusters that have identical sequences, and then we see the accumulation of SNPs in some patients but not others that we know are linked to a particular outbreak. So there's a lot of research going into trying to accurately tune how closely related is related and how many mutations we can see. The bug has a molecular clock, and that's what we expect it to mutate. So around 2 mutations per month is kind of around what people estimate that this bug will evolve. but those kind of really fine level differences, I think we're still a little while away from. For example, at the moment, we're giving you what's called a consensus sequence, which is a sum of all the different types of viruses that are in that particular patient. And so there's been really interesting suggestions that you could look at these like small minority populations to try and help tease out these transmission chains. That's technically really challenging and something that we're trying to actively collaborate and research and do some research around.
Yeah, I think that idea of microevolution and how it actually impacts on the way we use the data is really important for us to understand as well. And I think it's a lot of people don't sort of understand mutation and strain and how all those things happen. But this idea of a molecular clock is really important, and it does vary according to different viruses. And so some will have much more rapid error in transcription than others. And so this is one that's relatively stable, even though you will pick up these minor differences. But if you think about an infection, which is the result of the virus replicating in our bodies, this is billions of viruses replicating, in a small cluster, let alone, or a person, let alone, at a population level. And every time you get a transcription, there's a chance that you'll get this flip in those letters in that, RNA code that the virus has. So in that sense, when you think about it, with the amount of COVID there is in the world, you do expect variation, but we're still not seeing the big changes that can happen with those that have a very, unreliable replication process. And so that comes back to the point about, we do not want to see this become more infectious. We do not want to see this become more virulent or more dangerous. And they're the risks you have if you have, high levels of mutation happening as well. So in some ways it frustrates us because you don't always get the resolution, or you're getting these fine, detailed shifts in the genome that are hard to understand, unpack, and to use in a way to assist our outbreak control. But at the same time, it's a good thing that we don't have these big changes. And that will also matter for vaccine. And the stability of the virus, gives us a greater chance that the vaccine, particularly if it's targeting very stable areas in the virus RNA, like those coding for the protein spike, gives us a chance of finding a vaccine that might, in fact, as long as that immunity is durable, cover us for some period of time, not something you have to change all the time, like we do with flu vaccine.
We've got a question here from Andrew Leeson from Agence France Press, and he asks, the question's for Rebecca. Are you able, although I'm sure you could also help with this too, Catherine, are you able to give us an idea what the genomic testing teams and contact tracing teams go through just day to day to uncover the source of an outbreak, perhaps using a past example?
Sure. So there's probably around 10 people at the moment that are very busy in the lab. So once the sample comes into the lab, it's tested using a diagnostic test, and that gives us the yes, no, you have COVID-19 or you don't. Once we get that yes answer, the genomics team will grab that sample, and then they'll do what takes about 5 days. So it's a reasonably lengthy process to get the sample, to extract all the nucleic acid, in this case, all the human microbial and also viral nucleic acid out of the sample. And then we amplify the virus in small pieces to try and, so different genomes have different sizes, and a virus genome is very, very small. So it's kind of like a needle in a haystack analogy. So you have to look at a lot of pieces of hay until you find the needle, which is the virus. And so we use strategies to amplify the amount of needles, or get out our metal detector, if you like, to pull that out. And then we conduct the sequencing, and that alone on the sequencer takes 24 hours. So it's quite a laborious and lengthy process to actually go from collecting these samples to the endpoint. And there's a lot of wet lab work that needs to be done. And then there's a lot of computational biologists that are sitting at the end, and they're really analysing this code and the variations with this code with different mathematical models, and they predict the family tree. And we kind of work in parallel, if you like, with the contract tracing team. And then once a week we kind of meet and we independently cluster them in New South Wales using the genomics. And then we have a discussion about how the genomics and the epi kind of work together, and what are the cases that they're surprised about and which are kind of supporting each other, if you like. So it's quite a laborious kind of time-consuming process, and that's why sometimes you can see these delays in the results where they come out and talk about an outbreak that might have been released in the media, 5 or 6 days ago.
So, and I think that's right. It has to happen in parallel because it's so critical that the contact tracing happens, with the first ring of contacts within 24 hours of a result coming in. And in New South Wales, they're in a good position because they're actively, chasing these smaller clusters. And so you hope to get testing earlier in the process. So when someone's just got symptoms, or where someone is, a contact who's already become positive, but they've been tested possibly, even before they've got symptoms. So actually getting that work done, pulling together all the long form information. So the teams, which can include epidemiologists, will ask about quite detailed questions about what people have been doing, where they've been, who they've been in contact with over the preceding, usually 14 days. That takes you back through the period where they might have contracted the virus themselves, and also into that period before the test where they might have also been infectious for other people. So it's quite complex information, but it's critical that that happens quickly, because you'd be going out as an epidemiologist to the contacts and asking them the same questions and testing for, sending them off for testing, but also checking for symptoms and so on, and possibly going to their contacts before you even get the results in. So 10 days down the track, and you finally get the genomics, you've probably potentially gone through 2 or 3 generations of cases. And in fact, we know that in New South Wales, in the follow-up from the Crossroads Hotel, that by the time, and it was only 10 days after the exposure in Crossroads Hotel, when they got to contacts, they were already positive. So you have to do these 2 things in parallel, but it's fantastic having this process of meeting weekly and cross-checking your information and looking to see if anything's, saying, actually we assumed it was part of the same cluster, but it's got different genomic fingerprint. All of those things are really powerful ways to get the most out of, both branches of this important, contact tracing, virus tracing process.
We've got a question here from Claire Petty from the Adelaide Advertiser. I'm not sure if either of you two will be able to answer this, but we'll definitely throw it out there. Is anyone doing this work in South Australia, and do you have anything to share about the results from South Australian infections, such as where did the Barossa cluster come from, for instance?
Sorry, most of these investigations at the moment are pretty state-based. So I know that there's sequencing going on in South Australia through kind of knowledge of the public health laboratory network, but I'm not privy to understanding any of the kind of local context in South Australia, unfortunately.
Yeah, and nor am I, but I do know that they do have really good working relationships between their Health Commission and the laboratory systems over there. And oftentimes when they are talking about associations, it's not always clear whether it's based on genomics or it's based on good old shoe leather epidemiology, but where you have access to genomics, they would be using both together. So I think it's probably coming through in what they're telling us about relationships. If they're not telling us, it means that, if genomics hasn't been considered, then I'm sure they would be doing that in parallel if they wound up with unknown source clusters or cases.
Are you aware at all, Catherine, on what transfer there has been between South Australia and Victoria in terms of case numbers or cases going from one state to the other?
No, look, I haven't heard anything except that I think essentially we're saying that Australia had suppressed community transmission to the point of 0 or very low numbers that have since died out, and that we're looking at related strains now to the breach in Victoria wherever we have found cases. So unless it's a traveller who has tested positive late in the piece or some other imported virus, essentially we're working on the assumption, at least I am, and I'm sure that's what we're learning from the hotel investigation as well, that we now consider this to be a true second wave, in that it started with a late May introduction of the virus to a family that then tested positive and probably had 2 episodes of that across 2 hotels that then, fed it into the community. So I think everything that's not a new import and a traveller-associated case is linked back to these.
So as Australia continues to control the virus, we've seen Victoria's cases are lowering. What role does genomic testing play once the cases are sort of under control, or we've technically or eliminated the virus, such as we saw in New Zealand prior to their elimination ending, so to speak?
Well, it's very much about the fingerprinting, and Rachel can talk more about that. Rebecca can talk more about this too. But I think, at the end of the day, it's if we do get down to a very few cases, then we would be following that epidemiological investigation with the genomics as quickly as we possibly can to make sure that it wasn't a new lineage that we hadn't seen in Australia before, showing us that something had been reintroduced. So in a way, you don't want to see more cases, but if you do, you at least want them to be with a familiar lineage so that you can at least be reassured that you haven't got another new incursion of the virus into community that could be taking off. You're still suppressing the last, segments of community transmission.
Yeah, I agree with Catherine. I think we're really focused on looking at all the kind of importations. So we look at the cases that occur in the quarantine hotel, particularly what happened in Melbourne. It could happen to anyone. And, as reported by New South Wales Health, we have seen it happen in New South Wales. And it's really not about, unfortunately, these things are going to happen with this really transmissible virus. It's about trying to understand them as quickly as possible and then understand how we can live mission with that increased data and understanding of how the virus moves in certain situations. And really that's our focus at the moment. We would like to sequence more genomes. There's some limitations around the science of we need cases to have a significant viral load. And at the moment that's great because we have so many people coming forward early when they have symptoms, and I think we're starting to see a better recovery rate from those people coming forward. We're also using genomics to look back at different situations that occurred during the first wave and to understand, what infection control policies worked, what didn't work, what environments should we minimise, what situations can we really limit so that we don't see transmission. And there's a lot of talk about kind of super spreading events and defining what they are and what the risk factors of those type of events are, I think is kind of paramount at the moment.
We've got a follow-up question here from Andrew at AFP. He asks, in such an intense, laborious process, but under that pressure of a pandemic, how do teams feel when they make significant wins, like successfully tracking the source of an outbreak?
Yeah, it's pretty rewarding. I guess there's a lot of work that goes into it, but when you see that work is making a difference, and I guess everyone's sitting at home kind of feeling quite helpless. And so I think there are so many scientists excited about being able to help and, using our skills in some way that could assist. And that's really what started, at least in New South Wales, the kind of real effort to sequence all the genomes. We knew that this might be important. We didn't know how fast the genome was going to evolve and how we could kind of integrate the data with the epidemiologists. But I think, it was just pure kind of let's help. This is not an ideal situation. How can we assist? And I think the kind of collaborative effort has been essential to minimising the spread in New South Wales and in Australia in general.
And it's great that we've always, I guess, been building towards these better integrated relationships. It's always been an important part of the public health outbreak control work, but I think, we've probably taken it to a whole new level now with these sort of high-tech whole genome sequencing as well as, rapid sequencing work that now is being done and more routinely done. I think, at the shoe leather epi point of view, I haven't been involved in the contact tracing or the analysis, and this is probably a different outbreak to the one that, a lot of us would experience in our working lives because of the scale of it and the fact that Victoria is multiple outbreaks. It's hundreds of new outbreaks a day, in effect, that turn into these household or workplace clusters. And now we're down to 4. So that's a very different world to be in and something that starts to look a bit more familiar to most epidemiologists. But it is that kind of high-five slap when you get that moment and you think, we've nailed it. We've actually got strong enough evidence now for the source of this infection, whether it's, I worked on outbreaks that were pertussis moving into parts of New South Wales when I worked with New South Wales Health that hadn't been exposed before, where you had babies at risk. And so when you can shut an outbreak down and you've protected lives, it's hard because you never see the people that benefit from it, but just knowing how important it is to stop transmission and prevented further cases is what epidemiology is all about. And so that's what motivates and drives us. And when you're involved in a hands-on way at any part of this step that helps contribute to shutting down outbreaks and knowing how to prevent the next one, that is so profoundly satisfying. But that moment when you discover it, when you do the analysis and you identify something that's a real risk, and all your ducks line up, your environmental testing, all those things show you that it's a consistent pattern and you can make a case, you've identified a new food source or you've identified a risk behaviour, whatever it is, you can then really make a difference. So that's what it's all about.
You both sort of mentioned there what this was like compared to other outbreaks that you've worked on or other sort of infectious diseases. To give the scale of COVID-19 and its, huge impact, how does this compare to, say, Catherine, your epidemiological work with other infectious diseases, say the flu or pertussis, as you mentioned? And same with you, Rebecca. As a virologist or genomic tester, have you worked on other infectious diseases that just pale in comparison to COVID?
Yeah, look, as soon as you're talking pandemic and you've got an issue with transmission across borders, which we had initially, it's a very different world to where you're looking at a local outbreak of an endemic, condition that we're used to dealing with on an annual basis. It has multiple factors that make this different, like SARS originally, and, it's actually not knowing the organism. So we had like in SARS, we actually had a full genomic sequence before we understood how it was transmitted. And that's the challenge. We learned from SARS, so it didn't seem as scary in many ways epidemiologically, looking at the COVID-19 or the SARS-CoV-2 virus that causes the disease. So there were some things that were a bit familiar, but you're still really starting from that baseline. You've got to start doing, as Rebecca's already talked about, building up the genome. The Doherty Institute in Australia was the first to culture the virus in the lab, which then enabled this genomic work to be done and shared with the world. And those steps are very important, but you've got to have it hand in hand with the basic epidemiology to know how to protect. So you always start with the basics. Hand hygiene is where we start with every single communicable disease outbreak. It always works. It does no harm. But in fact, it's then building on that and understanding what puts parts of society at risk, who's going to be the most vulnerable in terms of serious illness, and piecing together the epidemic story and the vulnerable population story at the same time as, Rebecca's can tell us about how you start from scratch. So having an unknown organism changes the way you approach an epidemic completely. Having it on scale globally and knowing that you can't control the world, you can only control perhaps your area, is difficult before you shut borders. But even within Australia, now this second wave in Victoria, the sheer scale of it takes you to a whole different level of containment. So the big thing is that we kept our contact tracing going, even though we were getting, high 700 cases a day adding to the mix. And I know there's been concern about contact tracing and always wanting it to be bigger and better. But when you work at that scale and you're trying to link the information behind the information that's collected, it's so much more difficult to manage the analytics that help you, do that cross-matching between cases, tie them together and get, more linked cases that you can then understand transmission chains. So the scale actually changes the epidemic as well. And as I said, we had multiple epidemics in Victoria and still do at the same time. And so you have a situation where higher levels of community transmission seed more workplace outbreaks. They grow cases very quickly across the workforce. They, before that's discovered, feed it back into the community. And you get these accelerations in your epidemic that are happening in different parts of your community. that are hard to tease out. So again, genomics can help you do some of that, but the sheer pace, the speed of this and the size of it just challenge you to even keep up with it when you're talking about, secondary cases sometimes, coming into the picture when you haven't even had 48 hours from the first, and you might not have heard about them until you hear about the two of them at the same time.
And I think from a lab view, I work in a very, very busy kind of central laboratory. So we're not just exposed to the genomics, which was a whole kind of new bug, new pipeline, both wet and dry lab that we were kind of developing on the fly. You're kind of within the diagnostic laboratory, and I think they probably don't get a big enough pat on the back, because for the last kind of 7 months, the kind of leading story is how many tests did we do? And so they are just running at 300 times capacity the whole time. And so that's an enormous effort on the back of more specialised testing that's going on the back of building capacity, like Catherine said, to grow the virus. And that allows us to do a lot more kind of wet lab scientific type testing about this bug. And it's just, the size and the scale of building those systems back up, and the amount of kind of pressure and the, interest from the general public as well about what's going on. So balancing all those kind of things out is difficult. And from a genomics point of view, taking this quite complex data and simplifying it, so an epidemiologist who's got maybe 20 minutes in her day to try and figure out his day to try and figure out how these cases link in, and integrating that information is really, really challenging. Everyone's under time pressure, and it's kind of, how can I simplify this story so that they can take this bit of information and integrate it back into their system. So it's been an enormous effort by a lot of people. And I think, what we've done in Australia has been really impressive, I think, and the scale of both diagnostic testing and genomic testing and the work from the public health units doing all this contract tracing has really put us in this position, quite enviable position, where, yes, we do have this second wave, but it is incredibly minor compared to what we've seen overseas.
And I think also, and I think that's a really good summary, because I do think there's a lot of unsung heroes in here. And now that this has been running, even though we had a bit of a lull in between, but, 6 months quite hectically, that we're in a situation where, there's a different kind of challenge in keeping this going at the pace that we've been doing. But I do think, the public as well, who've been coming forward for testing, not those that are contact traced or part of a workplace outbreak or whatever, but the people with symptoms coming in, the government pushes hard to get people to keep doing that. But in fact, we've had quite low positive rates in Australia compared to other parts of the world, which shows us that we do still have people actively coming in to test. And we want low positive rates, but we also need to have a lot of people getting tested to be sure that is reassuring and telling us about what's happening in community. So it goes right back to the people making that decision about whether they go and get a test or not. And it's all of those steps working together that gives us the best chance of controlling this, but also sets this up as a good model for other countries to help, should they be facing these second waves, to try and cut them off earlier than they did the first time. We were very successful the first time. We didn't do as much testing. We didn't do any testing in the community unless they were linked to known cases, which was unusual. And so, in fact, we don't really have a good measure of what we did the first time. We do know the second time, particularly the different stories from New South Wales and Victoria, and the world is learning from that. They are very interested because it helps us, figure out where the pressure points are, where our weak points are, what you need to do to contain this so we can live with it for long enough till we get the vaccine.
I've got another question here from Claire Petty at the Adelaide Advertiser. This one will be for you, Rebecca. Is there any evidence to suggest the Hillsong Conference in Sydney was a source of COVID cases in Australia?
I don't really get that level of data to even postulate that. So we are not privy to kind of locations and things like that with the genomic sequencing data. So anything, I must say that the public health response and what's been communicated from New South Wales Health is incredibly transparent, and they're really making the hotspots well known and communicating that back to the general public to aid, people coming out and getting tested. But again, we're really not interested in kind of, highlighting particular segments of the community that might have unfortunately had more cases than others. But yeah, we're quite blinded to that. And I think one of the strengths of our investigation is that it's quite independent. We're not swayed by what the epidemiologists think. We think, these are genomic clusters, but for us, they're genomes. They're not kind of cases that are linked to all this secondary data that Catherine's talking about. But I think having that two-pronged approach, having a very kind of analytical science kind of yes-no binary output is kind of good when you're looking at these very complex subjective data sources as well. And I found that really useful.
We will wrap up shortly. So if anyone does have any questions, we will ask them, get them in now into the Q&A box in the bottom right. We have a call-in user too. Sorry, I've just had another question come in through to me. Bear with me. So we've got a question here from Jason Gale, and he asks, can we use serology? Oh, hang on, let me get my words right. Can we use serology to understand phylogenomics historically? Thanks for that question, Jason.
You go, Rebecca.
We're looking at two completely different things. So serology is looking at the body's ability to recognise a pathogen after it's happened. So once you get infected, then the virus is running through your bloodstream and you will make antibodies to that particular virus, and they will remain within your blood and can be stimulated again. And so serology looks at your body's response, or positive or negative, so whether you've got antibodies or you don't, as a measure of whether you've been infected. One of the big measures is whether you've been infected. So it doesn't really tell you very much about the virus, and phylogeny tells you about the virus and how it's evolving. So I think, you kind of, serology is looking at the host and phylogeny is looking at the virus itself, if that is clear enough. Maybe Catherine can add some commentary on that.
No, that's exactly right. You're looking for evidence from the human response that the virus was present, but the virus is no longer present. So you can't isolate the virus, and so Rebecca and her team can't do their good work because the virus is no longer present. And there are other situations where, they can pick up historic infections because they can still pick the virus up on PCR, but the virus is essentially dead. And so you can't culture it and do some of the work Rebecca's talked about, though you can do enough to identify the virus, and you might be able to do some work. I don't know if you want to comment on that, Rebecca.
I think serology. I think serology is really important, and it's just like we're talking about. You want to get out your toolbox and shake out all the tools that we've got to try and understand how this plays out. And so serosurveillance, going back and having a look at, we found that, or we've diagnosed this many infections, but whether actually more in the community, that's something serology can tell you. It can tell, it's been used, I know, in our laboratory to look at these cases where there are really low levels of virus in their sample, and they're really worried about there's a single PCR being used. So it adds evidence to suggest, they've got a low-level virus, but they've had a previous infection. And that helps the epidemiologist to go back and go, okay, this person was positive, where were they, who have they had contact with, let's isolate all those cases. So it's another tool. It's not a tool that I can use because I'm interested in the virus itself rather than the host response, but it's incredibly important test to use to understand retrospectively what's happened in the community.
But Rebecca, if you tested someone, and we've had these situations where they were positive on a PCR test, so we've found, virus in the back of their nose or throat, and then they clear and you get some negative tests, but you find a positive test later, and we think that's not a reinfection. We think that's probably still just dead virus RNA, in the system. Can you actually look at that virus genome and see if it is the same virus that was responsible for the first infection, if it's dead, and you can't culture the virus off the swab, but you've got the virus RNA on the swab?
Yep, so we don't culture the virus to do the genomics. We just go straight from the clinical sample. So if there was enough virus present, and unfortunately you have to have enough copies of the virus, then we could amplify that virus and look at the genome from point A and point B. I think at the moment, because the virus within the community is so similar, it would be hard it would add evidence, but you couldn't say conclusively that they hadn't been reinfected. But yes, in those cases where they've had a positive and then they kind of had this dribbling kind of inconsistent negative results, it's definitely suggestive that they've got this kind of residual leftover virus. And I know that they did a lot of work with culture. So they would take those cases and then they would put them into a laboratory culture system and make sure that those people, that they couldn't re-inoculate the culture with that. And that happened extensively with the first cases that we saw, because we wanted to make sure that when we released these people from hospital, they'd recovered and they weren't able to transmit the virus. But we knew a lot less about, the length of the infection and things like that in the very early stages in late January.
Yep.
We've got a call-in user, so we're just going to unmute them just to check if they have any questions. So just bear with. So person on the phone, you are now unmuted if you have any questions for our speakers.
I'm assuming that's me. No, I'm fine. Thank you.
Not a problem. Thanks very much. I've sort of got one final question. So if anyone does have any more questions, get them in now, because we will be wrapping up shortly. And so I guess probably a bit of a final question just for the both of you, as you're in different states. How do you see your states going from here? I haven't actually seen the case numbers for today, Catherine, but how is Victoria looking to you?
Look, we're very much in a consolidated, improved, pattern of downward trend. Our number of new cases today is 240, so it's consistently now in the 200s. And I think just when I was looking at our numbers, our 5-day average has dropped from 360 last week and 500 the week before. So we're actually really consolidating this now. The big news, though, as I alluded to earlier, is the fact they've announced we only had 4 new outbreaks yesterday. And that's a real sign that we're shifting the fundamental dynamics of the pandemic, and that we now have quashed that community transmission sufficiently that it's not being taken into either new clusters, new households or workplaces to start these, rolling, increased case numbers continuing. So that will allow us to see the numbers come away. But in particular, we're still expecting this lag in terms of having so many people still in hospital, and while that's levelled out, now it's plateaued, we still do have a lot of, active situations where people are, in some cases, fighting for life. And so we still have somewhere in the mid-40s, the number of people in ICU, and until those numbers really start to come down, where those people aren't being replaced when they recover and leave hospital, hopefully, then we're still going to see our hospitalised numbers stay high. But in particular, there's another lag then to the people who lose the fight. So we have to be prepared for a few more days where we could be seeing high numbers, but those will also all start to come down as our active case numbers come away and we don't replace them with new cases, and we stop this terrible dynamic of feeding outbreaks. So that's a fundamental shift in the dynamics of the epidemic for Victoria that I think, start to bring us back into a world that, is still more active, but starts to look a little more like the New South Wales control that's possible when you get the numbers down.
Yeah. Annika, as far as New South Wales, I just looked it up. We've only got 5 new cases today, so I think we see these are kind of under 10 numbers, and I think none of those cases are kind of unknown sources. So I think I'm pretty optimistic about the situation in New South Wales, and I'm hoping that it continues to kind of just, we reduce those numbers over and over the next couple of weeks, and we get back to a more comfortable position in terms of number of cases that we're seeing per week.
And I guess one final question. We haven't had any other questions come in, so this will indeed be the last one. But how do you think the COVID-19 pandemic has contributed to epidemiology and genomic testing? Has it led to an advancement that if we have future outbreaks, we are, much better placed than we were, say, at the beginning of 2020?
Rebecca, did you want to go first?
Yeah, I think definitely we already had pretty strong links to the communicable disease branch in New South Wales as far as the genomics team goes. But I think we'd have a lot more confidence and a lot more kind of open strategies for implementing a similar kind of system if there was another pandemic. I hope that it's another 100 years before we see a similar situation. But I think that genomics is probably going to be brought on board more and more for different infectious diseases in the future. And it's really shown how the culmination of genomics and epidemiology is really important in stopping transmission. And Catherine used the whooping cough example, and I think that's excellent as well. Those kind of bugs are still problematic in the community. They don't get as much exposure as obviously a pandemic does. So we don't usually talk in the media about them, but infectious diseases in Australia still exist, and we want to. Our job is to minimise them and minimise the kind of influence of those bugs on the community, and understand how we can build better vaccines even and that's something that we use genomics for in the whooping cough example.
And I think from an epidemiological point of view, it is this idea that we have to work in teams, and we've always known that. Epidemiologists always do. And now I think, absolutely we have a much more mainstream approach to it, which includes genomics. And so that we have to work that way because it's such a powerful tool to add to our kit. In terms of epidemiology itself, it's interesting because over the years, I've been an epidemiologist since last century, but there was this sense that in the developed world in particular, that we were looking at communicable disease issues, and infectious diseases didn't require the same. We'd managed them. We had antibiotics, we had vaccines, and of course now we have new pathogens that we don't have vaccines for, and our antibiotics. And I do a lot of work in the antibiotic resistance space. Don't cover all the infections we can have now. We have a level of resistance that's turning into and has been recognised as one of our major public health risks. So the world's been testing a bit anyway, but we actually don't have a lot of expertise. So while the general public can now say epidemiology and actually have a bit of a sense of what you might do, a lot of it's still looking at, they think about it in terms of the numbers or the lab work, but it is these fundamental skills to understand, how you manage outbreak control on the ground, how you understand the numbers and all the things that sit behind the numbers that allow you to make sense of them. That this is a really critical skill area that I think we've probably identified we don't have enough capacity. So as we're growing capacity in our contact tracing, I think we also will grow more expertise in some of these areas that will increase our surge capacity in these situations. But also I think it's the way we work nationally, and the National Cabinet is one of those great innovations that really helped us work collectively and have a more consistent approach to managing this and also communicating around COVID. And so I think actually having that ability to work together across states, finding ways to really support that, whether it's a CDC approach or not, that you actually have some sort of central way that then supports the states in what they're doing. And we've seen that Victoria's had a greater need than anyone else, in the last month. But also you bring together that expertise, so you've got this capacity and you're working off the best information, and you're setting things up so they're streamlined between the country and the states. And we've moved towards that during the outbreak, but some of that, we weren't ready for this in the same way. So there's so many different levels to this that we've learnt so much, but it also is now giving us impetus to invest in these things. That's a really important lesson out of this, that we will take forward and actually be safer for that.
I think that's a lovely place to end, so we will wrap it up there. And so a full recording of today's presentations will be posted on SciMex very shortly. Feel free to contact us direct at the Science Media Centre if you have any specific requests or questions. We're on 0871 208 666, or you can email us at info@smc.org.au. Thanks again to Rebecca and Catherine for joining us, and thanks everyone for joining us today for this briefing.
Thank you.
Thank you, Annika and Catherine.